Welcome to this lecture on the principles of ecology. Today, we are going to explore two fundamental concepts that dictate how life spreads across our planet: dispersal and dispersion. While they sound similar, they describe very different ecological processes. Let us dive in and understand how populations move, why they leave their homes, and how they organize themselves in nature to maximize their chances of survival. To begin, let us look at ecological dispersal. Imagine growing up in a crowded house. Eventually, you need to move out to find your own space. In nature, dispersal is this exact journey. It is the one-way movement of individuals or their seeds and larvae away from their parent population. Unlike a daily commute or seasonal migration, dispersal is a permanent relocation. It can be active, like a young wolf seeking new territory, or passive, like a dandelion seed blowing in the wind. This journey always involves three distinct phases: leaving the original site, traveling through the environment, and finally settling in a new home. Now that we understand why dispersal is, we must ask why it matters so much. Dispersal is absolutely critical for a species' long-term survival. If organisms never moved, they would quickly eat up all local resources, leading to starvation and population collapse. By moving away, they reduce competition and avoid inbreeding. Dispersal also allows species to colonize new areas, such as a forest recovering from a fire or a newly formed volcanic island. Most importantly, it mixes genes between different populations, maintaining the genetic diversity needed to adapt to diseases and a changing environment over time. This brings us to an important question. How do we measure dispersal? Ecologists often use a simple exponential decay model to predict how far a species might spread. The formula is n of x equals n0 times e to the power of negative d times x. Here, n of x is the number of individuals that successfully settle at a specific distance, x. N0 is the starting population at the source. The constant e is the base of the natural logarithm. Finally, d is the dispersal coefficient. If d is large, individuals do not travel far and the population drops sharply. If d is small, they travel much greater distances. Before we move on, let us see this formula in action with a concrete example. Imagine a mature oak tree producing 1,000 acorns with a dispersal coefficient of 0.05 meters per meter. We want to know how many acorns will settle 20 meters away. We start by plugging our given values into the exponential decay formula. Multiplying the coefficient by the distance gives us an exponent of -1. Using a scientific calculator, e to the power of -1 is approximately 0.3678. Multiplying this fraction by our initial 1,000 acorns gives us about 368. This steep drop-off highlights how difficult long-distance dispersal is for plants. Now that we have explored how organisms move, let us look at where they end up. This is called dispersion, or spatial distribution. If dispersal is the dynamic action of moving, dispersion is the static pattern of where individuals actually live. Imagine looking down at a city at night. Houses are not scattered randomly, but clustered in neighborhoods or lined up along streets. Similarly, taking a snapshot of a population reveals its dispersion. Understanding this pattern is incredibly important because it immediately reveals the underlying ecological forces at play, such as animals fighting over limited resources or hunting together in packs. This concept of spatial arrangement leads us to categorize dispersion into three primary patterns. The first is clumped dispersion, where individuals group together in patches. This usually happens because resources like water are unevenly distributed, or due to social behaviors, like wolves in a pack. The second is uniform dispersion, where individuals are evenly spaced apart. This is almost always the result of intense competition or territorial behavior, like desert plants fighting for scarce water. The third and rarest pattern is random dispersion. Here, individuals are spaced unpredictably, which only happens when resources are consistent and individuals barely interact with one another. To truly grasp these patterns, let us look at this diagram. On the left, you see clumped dispersion. Notice how the individuals, represented by dots, are tightly gathered into distinct groups, leaving empty spaces between them. In the middle panel, we have uniform dispersion. Every dot is kept at a strict, even distance from its neighbors, perfectly illustrating territorial boundaries. Finally, on the right, we see random dispersion. The dots are scattered completely by chance, with some close together and others far apart. This visual snapshot is exactly what ecologists look for when studying a new habitat. Now that we can visually identify these patterns, how do we measure them mathematically? Ecologists use a tool called the variance to mean ratio, or VMR. They divide the habitat into equal grid squares, count the individuals in each, and apply this formula. VMR equals S squared divided by X bar. Here, S squared represents the statistical variance, which shows how much the counts fluctuate from square to square. The symbol X bar represents the mean, or the average number of individuals per square. By dividing the variance by the mean, we get a precise index of dispersion. This formula we just saw leads us to a set of established ecological rules for interpreting the result. If the calculated VMR is exactly equal to 1, it indicates a random dispersion pattern because the variance perfectly matches the mean. If the VMR is significantly greater than 1, it means the variance is high. Some squares have many individuals, while others have none, indicating a clumped dispersion. Conversely, if the VMR is less than 1, it means every square has roughly the same number of individuals, which points to a uniform, evenly spaced population. Let us apply this mathematical tool to a real-world scenario. An ecologist counts rare orchids across 10 forest plots. The statistical analysis gives a mean of 3.8 orchids per plot and a variance of 34.4. We want to determine the spatial pattern. We substitute our variance and mean into the VMR formula, setting up a simple division of 34.4 by 3.8. This gives us a variance to mean ratio of approximately 9.05. Because this number is significantly greater than our baseline of 1, we can confidently conclude that these orchids grow in a highly clumped pattern, likely tied to specific favorable microhabitats. To wrap up our lecture, let us review the most important ideas. Dispersal and dispersion are fundamental yet distinct concepts. Dispersal is the dynamic journey of individuals moving away from their origins to find new resources and maintain genetic diversity. Dispersion is the static result of that journey, the spatial arrangement of individuals once they have settled. Whether a population's dispersion is clumped, uniform, or random, it beautifully reveals the underlying environmental resources and social interactions at play. Together, these concepts explain how life successfully spreads and organizes itself across the globe.